When you think of microbial life cycles, what do you think of? Probably mitosis, asexual division, binary fission, possibly budding or lateral gene transfer. What about sexual reproduction? Diatoms are microbial algal organisms that can reproduce sexually, but what makes them unique is that diatoms alternate between asexual and sexual reproduction. It has long been thought that sex was an inevitable consequence of the unavoidable reduction in size that happens when diatoms mitotically divide, and that diatoms simply use sex to restore maximum size. But what if size reduction is not unavoidable, but rather a selective advantage that allows diatoms to time and maintain sexual reproduction? Diatoms, also known as Bacillariophyceae, are a monophyletic group of photosynthetic algae, belonging to the Stromatopile clade of the SAR supergroup. They evolved between 160 to 240 million years ago during the Mesozoic era, and have since diversified into over 250 genera and as many as 200,000 species, making them by far the most species-rich group of algae. Diatoms can live in almost any aquatic or semi-aquatic habitat and are extremely productive. They count for about 1/5 of yearly production of oxygen on Earth, 1/2 of total marine carbon fixation, and contribute almost 1/4 of total global primary productivity. Diatoms also play an essential role in the biogeochemical cycling of silica because of their unique silica-infused cell walls. These glass houses are structured like a petri dish, with two parts, a bottom and a larger top that fits snugly over the bottom. Each half of a diatom cell wall is called a theca. The smaller half is called the hypotheca, and the larger half is called the epitheca. Each theca, in turn, is composed of two parts, the valve, which forms the majority of the theca, and the girdle, which is made of circular bands of silica attached to the edge of the valve. The girdles hold the two theca together. The entire diatom cell wall is called the frustule. The separation of the top and bottom theca is what allows diatoms to divide during mitosis. To prepare for asexual division, the diatom cell elongates and the theca are pushed slightly apart. Each daughter cell inherits one of the parent's theca, which becomes the epitheca in each new cell. This is because the process of division occurs within the rigid parent frustule and is thus constrained by the size of the parent. Unable to make a theca bigger than the parent frustule in which they are enclosed, each daughter cell will form a new hypotheca to fit within the inherited theca. Only then are the two new cells exocytosed from the parent's now empty frustule. From this process, one daughter cell will be equal in size to the parent, and one will be slightly smaller. Over many generations of division, the average size of cells in a diatom population will decrease, and the standard deviation in cell sizes will increase. This is known as the MacDonald-Pfizer rule. When cells are as small as about 30 to 40% of the maximum size, they become sexually viable, and if environmental conditions are suitable, sexual reproduction can be initiated with another sexually viable diatom. Many types of sexual strategies have been documented in diatoms, including oogamy, a type of heterogamy, where the female gamete is a large, non-motile egg and the male gametes are small, motile sperm. Isogamy, in which both female and male gametes have similar morphology. Behavioral heterogamy, in which male and female gametes differ in function but not morphology, and even self-fertilization. Fusion of gametes creates a zygote, known in diatoms as the oxospore. The oxospore expands in volume and gives rise to the initial cell, which is the largest cell of the life cycle. The whole process of sexual reproduction lasts a few hours, and is again followed by months or years of asexual division. This process of sexual reproduction allows diatoms to restore their maximum size after the steady decrease in size that occurs in mitosis. It has been hypothesized in the past that this size reduction restoration cycle in diatoms is unavoidable, and simply a consequence of living in a rigid glass shell. However, in more recent years, some diatoms have been found with the ability to grow vegetatively after mitosis to avoid a decrease in size, to produce oxospores asexually, thus avoiding sexual reproduction, and even the ability to directly divide into two equally sized daughter cells, which occurs via a modified arrangement of the girdle bands. So the question we ask here is, if size reduction and sexual reproduction are avoidable, then why in most diatom species have these processes persisted through evolutionary time? Sexual reproduction comes with both costs and benefits. For microbial organisms, the costs of sex tend to outweigh the benefits, although it's difficult to quantify this. One of the biggest costs for microbes, and thus diatoms, is the lengthy time it takes to sexually reproduce, during which time there is no cellular production and much cellular material can be lost. During sexual reproduction, diatoms are also much more vulnerable to grazers because of the fragility and desirability of their gametes. There is the potential for failure to mate, which is extremely costly. There is also the possibility of producing offspring that are less fit than the parents due to recombination of what might have been a very fit parent genome. In all organisms, sexual reproduction is beneficial because of the potential that genetic recombination could lead to a more fit offspring. Thus, sexual reproduction allows for faster natural selection and adaptive radiation responses to changing environments and ecological interactions, such as keeping up with the evolution of parasites. In microbes, sex can also reduce deleterious mutations that may accumulate as a product of asexual division. These benefits of sexual reproduction may still not outweigh the associated costs for microbes. Diatoms seem to have found a unique solution to this problem by balancing long periods of low-cost asexuality with short periods of beneficial but potentially high-cost sexuality. Thus, size reduction in diatoms may not be a passive byproduct of physiology, but rather a selective advantage that allows diatoms to time sexuality. This size-dependent mode of timing sex may be desirable over relying on environmental triggers, which generally cause a necessity for exactly once-a-year sex. By relying on size as a trigger, diatoms can space sexuality over intervals much longer than 1 year, which is beneficial for a microorganism whose sexuality costs may be high enough so as to warrant sex that is less frequent than 1 year. In this way, diatoms have found a way to minimize the costs of sex while still reaping the benefits. Other hypotheses for the persistence of this strategy are that diatoms simply have not been able to evolve a way around either size reduction or sex as a means of combating size reduction. However, the existence of a few diatom species which do have the ability to avoid these processes seems to prove that any diatom lineages is in fact capable of involving ways around the size reduction sexual reproduction strategy. Thus, size reduction is likely a product of natural selection because of the edge it gives diatoms in their ability to time sex and balance the costs and benefits of both sexuality and asexuality to meet their needs. The extreme species richness, diversity, and abundance of diatoms is evidence that this strategy has been nothing but extremely advantageous.